Welcome to another lecture by MedicoMedics, Cell Biology, the complete lecture series. Here is your complete roadmap to mastering cell biology, from the basics of cell structure to clinical applications and cancer biology. Stick with this and you will build a rock-solid foundation for everything that comes next in medicine. So let's get started. Welcome to another lecture by MedicoMedics, Learning Made Easy. Cell Biology, Chapter 1, Introduction to Cell Biology. So in this lecture, we will discuss the importance of cell biology in medicine. We will compare prokaryotic versus eukaryotic cells, discuss the history of cell biology, respond to the question why we study cell biology in medicine, and then end with a summary. Now, what is the importance of cell biology in medicine? Well, cells are the smallest unit of life, responsible for all life processes. Now, what's the relevance of cell biology in medicine? Well, understanding cell biology is critical for diagnosing, treating, and preventing diseases. For example, when we talk about cancer, we're talking about uncontrolled cell growth. If we're talking about another pathology like diabetes, we're talking about dysfunction in insulin-producing cells. Whenever we're talking about infectious diseases, we're talking about pathogens invading and hijacking our cells. Furthermore, cell biology drives innovations like gene therapy and stem cell treatments. So our cells are like bricks of a building. Understanding how they work helps us figure out what's wrong when things break down. So now let's explore the core concept that forms the backbone of cell biology, namely cell theory. The cell theory. Now, all living organisms are composed of one or more cells. From tiny bacteria to complex humans, life relies on cells. Furthermore, the cell is the basic unit of life. It performs all essential processes like energy production, growth, and repair. Furthermore, all cells arise from pre-existing cells. Cells replicate themselves to maintain life and pass on genetic material. Now we have two main types of cells when we discuss cell biology. We talk about prokaryotic cells and we talk about eukaryotic cells. So prokaryotic cells are simpler, smaller, they lack a nucleus and have minimal organelles. The DNA in prokaryotic cells float freely in the cytoplasm. An example of prokaryotic cells are bacteria. However, eukaryotic cells, so human cells, are more complex. They are larger and have a nucleus with organized organelles like mitochondria and the endoplasmic reticulum. These are found in multicellular organisms like plants, animals, and humans. Now you can immediately notice some key differences here. Prokaryotic are smaller, eukaryotic are larger. When we talk about bacteria or prokaryotic cells, they lack a nucleus, while eukaryotic cells have a nucleus. Where one has minimal organelles, the other one has organized organelles, including, for example, mitochondria. Now let's delve a bit deeper into the eukaryotic cell. Now, the eukaryotic cell consists of a nucleus, first and foremost, which is the control center housing the DNA. You can see it here at the very center. We then have mitochondria, which is the powerhouse of the cell generating ATP or energy. And you can see the mitochondria illustrated here. Furthermore, we have ribosomes. These are the factories that produce proteins. We have the cytoplasma, the workshop where chemical reactions occur. So this is the cytoplasma, right? All these organelles are floating in this cytoplasma. Furthermore, we have the cell membrane, right? The outer layers of the cell. This is the quote-unquote security gate regulating what goes into the cell and what goes out of the cell. So think of a cell as a busy factory. Now, each organelle is a specialized department, keeping the factory running smoothly. Now that we have an overview of cell structure, let's examine how cell biology has evolved as a field. So when we discuss the history of cell biology, a couple of names come to mind. First of all, Robert Hooke. All the way back in 1665, Robert coined the term cell while observing cork under a microscope. Antoine van Leeuwenhoek discovered microorganisms and called them animalcules. Now, furthermore, the development of modern microscopy enabled the study of cellular structures in detail. Now, why do we study cell biology in medicine? Well, as we mentioned, first of all, we need to understand diseases. Almost all diseases, from cancer to infections, begin at the cellular level. Furthermore, innovation in treatments, insights into cells have led to advanced therapies like CRISPR gene editing and immunotherapy. It's important for clinical diagnoses. Blood tests, biopsies, and imaging often target cellular structures or activities. Now, some future frontiers in cell biology include stem cell research, organoids, and artificial tissues, giving us hope for medical breakthroughs. And that's the end of the first lesson, where we had a brief overview. Continue now to Chapter 2. Welcome to another lesson by MedicoMedics, Learning Made Easy. Cell Biology, Chapter 2, Cell Structure and Organization. In this lesson, we will discuss different types of cells. We will delve deeper into the nucleus, discuss the mitochondria, ribosome, the endoplasmic reticulum, present the Golgi apparatus or Golgi apparatus, present lysosomes and compare them with peroxisomes, present the cytoskeleton, discuss the cell membrane, and end with a summary. Now, when we talk about types of cells, like we mentioned in the overview in Chapter 1, we talk about prokaryotic cells and eukaryotic cells. And if you remember, prokaryotic cells were more simple, smaller, and they lack a nucleus. An example of a prokaryotic cell is bacteria. Now, the key components of prokaryotic cells is they have cell membrane, cytoplasm, DNA, and ribosomes. While eukaryotic cells, compared to prokaryotic cells, are more complex. They are larger. They have a nucleus and membrane-bound organelles. These are found in humans, animals, and plants. Now let's begin with the nucleus. Now, what is the structure of the nucleus? Well, it's enclosed by the nuclear envelope with pores for material exchange. It contains, so the nucleus contains chromatin, which is DNA and proteins, and the nucleolus makes ribosomes. Now, what's the function of the nucleus? Well, it stores genetic material, right? It houses DNA. Furthermore, it regulates cell activities by controlling gene expression. So the nucleus is like a boss's office where all instructions are stored and decisions are being made. Now let's look at how energy is created in the cells. When we talk about energy and the production of ATP, we talk about the mitochondria, known as the powerhouse of the cell. The structure is a double membrane. There is an outer membrane, seen here, and a folded inner membrane, also known as crista. Now, the mitochondria contains its own DNA and ribosomes. Now, the function of the mitochondria is to produce ATP through cellular respiration. And it plays a key role in energy metabolism and in programmed cell death, apoptosis. Now, in later courses and lectures, especially in biochemistry, we will delve much more in detail into the production of energy in the mitochondria. But right now, all you need to remember is that the mitochondria are like power plants. These generate the energy that our body needs to function. Now let's move to the ribosomes, which are the protein-making factories of the cell. Now let's talk about ribosomes, or protein factories. Their structure is made of RNA and proteins. They are found free in the cytoplasm or attached to the rough endoplasmic reticulum, RER. The ribosome and their function is to synthesize proteins by translating mRNA. Proteins are essential for cell structure and function. So the ribosomes are like chefs in a kitchen. They are cooking up proteins based on recipes from DNA. And if you remember, the DNA is housed in the nucleus. Now let's talk about another very important structure, which is the endoplasmic reticulum. The endoplasmic reticulum is like a factory assembly line where raw proteins and lipids are processed and packaged. Now we divide the endoplasmic reticulum into the rough endoplasmic reticulum, or RER, and the smooth endoplasmic reticulum, SER. Now, the rough reticulum is covered with ribosomes. It synthesizes and processes proteins. And you can see the rough endoplasmic reticulum here, and the ribosomes covering it. The smooth endoplasmic reticulum doesn't have ribosomes covering it, and it synthesizes lipids. It also detoxifies drugs and stores calcium. Now, once things are processed here, where do these products then go? And to learn more about the next step, we will now visit the Golgi apparatus. So the Golgi apparatus, or the shipping center, which should make sense since the endoplasmic reticulum was described as the assembly line, right? We started with the nucleus containing DNA. We then had ribosomes reading the mRNA and translating proteins and producing proteins. These then continue into the endoplasmic reticulum, into the assembly line, quote unquote, and then arrive at the Golgi apparatus, the shipping center. So the Golgi apparatus can be thought of as the post office, where packaging and shipping of molecules are made and then decided on where to go. So the structure of the Golgi apparatus is that it contains stacks of membrane-bound sacs or cisternae. Its function is to modify, sort, and package proteins and lipids for transport. It also produces lysosomes and secretory vesicles. And the Golgi apparatus is illustrated here. Let's now examine the cell's recycling and waste disposal system, its lysosomes. So we've reached the Golgi apparatus where packing and sorting and shipping has been done. Now we need to have a system in place where waste is broken down, right? So lysosomes contain enzymes that break down waste and cellular debris. It also plays a role in autophagy, meaning recycling damaged organelles. Compared to that, we have the peroxisomes that break down fatty acids and toxins. It contains enzymes for oxidative reactions. So lysosomes can be thought of as the cleanup crew, and peroxisomes are like detox centers. Now, how do all these organelles that we have talked about, the ribosomes, the endoplasmic reticulum, the Golgi apparatus, how do these organelles stay in place? To find out, we need to talk about the cytoskeleton. Now, the cytoskeleton is the cell's framework. Its structure is a network of protein filaments, so microfilaments, microtubules, and intermediate filaments. What is the function of the cytoskeleton? Is to maintain the cell shape. It's to facilitate intracellular transport and cell division. Furthermore, another function is that it enables cell movement. So what the cytoskeleton does is that it provides the cell structure and furthermore provides transport routes. Now let's finish by understanding how cells interact with their environment through the cell membrane. We have now arrived at the cell membrane, which is made of a phospholipid bilayer with embedded proteins. It is dynamic and semi-permeable. What is the function of the cell membrane? Well, mainly to control what enters and what exits the cell. Furthermore, it facilitates communication through receptor proteins. Now, as we move on into further lectures, we will delve into details of how this cell membrane is constructed, how things from the outside actually enter into the cell, why things from the outside leave the cell, these different channels whereby things are entering, and also the benefits of having a phospholipid bilayer, because what you will learn is that the outer surface of the cell is hydrophilic, while the inner part of the membrane is hydrophobic. And this leads to different considerations to keep in mind. But more on that as we move on into further lectures. And now, in summary then, the cell is a complex structure with specialized organelles. Key organelles to remember from this overview lesson include the nucleus, ribosomes, the mitochondria, the smooth endoplasmic reticulum and the rough endoplasmic reticulum, Golgi apparatus, lysosomes and peroxisomes, cytoskeleton and cell membrane. And if you remember what sets them apart, in the nucleus is where we house the DNA. The ribosome is our protein factory. We have the mitochondria, the powerhouse where energy is produced. We have the smooth and the rough endoplasmic reticulum, where we have the synthesis of lipids and proteins. Furthermore, moving along into the Golgi apparatus, where we have the packaging, sorting, and shipping. And then we have the recycling part of the cell, the lysosomes and the detoxification centers in our peroxisomes. What gave the cell its structure and what helped facilitate transport routes? Well, we talked about the cytoskeleton. And then at the end, what is it that actually allows for things to enter into the cell or leave the cell? These are determined by our cell membrane, which was made by a phospholipid bilayer. Now, each organelle plays a distinct and vital role in maintaining life. Understanding cell structure is fundamental to diagnosing and treating diseases. Now let's move forward to the next chapter where we will explore how substances move in and out of cells, meaning we will go deeper into the cell membrane. And that's the end of this lecture. Continue now to Chapter 3. Welcome to another lecture by MedicoMedics, Learning Made Easy. Cell Biology, Chapter 3, Cell Membranes. In this lecture, we will discuss the structure of the cell membrane, discuss transport mechanisms. We will discuss passive transport, diffusion, osmosis, and facilitated diffusion. We will compare it to active transport. We will discuss endocytosis and exocytosis, cell membranes, and signaling, and end with a summary. Now, what's the structure of the cell membrane? In the previous chapter, we mentioned that it has a phospholipid bilayer. Now, the phospholipid bilayer is composed of phospholipids with hydrophilic, meaning water-loving, heads, and hydrophobic, meaning water-fearing, tails. It is arranged in a bilayer to separate the internal and external environments. Furthermore, we have proteins. So integral proteins span the membrane, acting as channels or transporters. Furthermore, we have peripheral proteins attaching to the membrane's surface. These are aiding in the signaling or supporting. Another thing is cholesterol, which provides stability and flexibility to the membrane. We also have carbohydrates that are attached to proteins and lipids. These are involved in cell recognition and signaling. And here in this illustration, you can see these different parts. So we have the phospholipid bilayer. We have the head here and we have the tails here. And as you can see, tails from the membrane wall on the outside of the cell and the membrane wall on the inside of the cell are connecting. So this is why we call it a bilayer. And we learned here that these tails are hydrophobic, so they don't like water, while the heads on the outside and the heads on the inside are hydrophilic. Another thing we learned is that there are proteins creating channels for things entering from the extracellular environment and things from the intracellular environment to exit. Now, there are other structures like carbohydrates and lipids, etc., on these cell membranes, helping with recognizing things that are floating by, wanting to enter, for example. Now, transport mechanisms overview. When we talk about transport mechanisms, we have passive transport, where no energy is required, and we have active transport, where energy is required. Now, when we talk about passive transport, we talk about diffusion, osmosis, and facilitated diffusion. When we talk about active transport, we talk about protein pumps or the process of endocytosis and exocytosis. So the main difference is that some substances will just sneak through rather easily through the processes of passive transport, and other substances need help or energy to pass the membrane. So let's dive deeper into passive transport first. So let's begin first with passive transport and the process of diffusion. Now, diffusion is the movement of molecules from an area of high concentration to low concentration. For example, the oxygen and carbon dioxide exchange in the lungs is an example of passive transport or diffusion. And again, no energy is required. So diffusion is like the scent of a perfume spreading across a room. It moves from where it's strongest to where it's weaker. Now let's look at a special kind of diffusion called osmosis. So another passive transport is osmosis. This is movement of water molecules through a semi-permeable membrane from low solute concentration to high solute concentration. This is very important in cells because it helps maintain fluid balance. Another passive transport is facilitated diffusion, where movement of molecules is down their concentration gradient with the help of a transport protein. So, for example, glucose transport into cells using carrier proteins. Again, no energy is required. So facilitated diffusion is like using a key card to enter a secure building. You still follow the rules, but you need special access. Now, what's important here is that we said the movement of molecules down their concentration gradient, right? Now, it is facilitated with the help of a transport protein. But what if we come across when the cell needs to move substances against the gradient? So when there is opposition and it's not enough to diffuse through the membrane, even with the help of transport proteins or by osmosis, for example. This is where we need to talk about active transport. So in active transport, we need energy to facilitate movement. And it moves substances against their concentration gradient. So for example, the sodium-potassium pump functions in this way. Another one is calcium pumps in muscle cells, for example. So active transport is like, imagine you're pushing a ball uphill, right? It takes extra energy to move things when gravity is pulling it back down. So this is where the role of ATP, and if you remember previous lectures, the main organelle involved in the production of ATP is occurring in the mitochondria of the cell. Two other important concepts are endocytosis and exocytosis. So endo means essentially in, while exo means out or exit. So endocytosis is the process by which cells engulf substances into vesicles. An example of this is phagocytosis, when white blood cells are eating bacteria. So you can see here in this illustration, endocytosis, right? Here is the bacteria, and it's being engulfed. It's entering into the macrophage, or the neutrophil in this case. And following along, it then degrades, or the bacteria is broken down. And when that is finished, it then undergoes exocytosis. So exocytosis then is the process by which cells expel substances in vesicles. Example is releasing the release of neurotransmitters in nerve cells. So just remember, endo, into, exo, or exit, is out from. Now let's discuss cell membranes and signaling. So we mentioned that there are on the cell membrane proteins, for example, that are receptors, and they are on the membrane detecting chemical signals. So for example, hormones or neurotransmitters. What then occurs is signal transduction. So signal binding triggers a response inside the cell. So here is an illustration whereby a ligand, which can be any molecule, here in blue, is binding to a receptor on the cell membrane. When it's binded, there is a relay, right? Signal transduction is happening, arriving into the cell, causing or creating a response. Examples of this in medicine could be insulin receptors for glucose uptake. Targeted drugs that block specific receptors in cancer therapy is another example in medicine where this is actually quite crucial. So what we do then is we take some drug to block this receptor so it doesn't bind here. Because let's say we find out that if this thing floating around binds to a receptor on our cell membrane, it will relay a signal leading to, I don't know, the production of something else, right? And we don't want this something else to happen. And since we probably can't get inside to kill these off, what we do is we say, okay, we know that this receptor is binding these. We don't want that to happen. So we will give a drug that will go and block this receptor. And then we will not have this response happening. So that's an example of targeted drugs that block specific receptors. And so the membranes of our cells act as well as antennas. They pick up signals and then pass them inside the cell to trigger actions. In summary then, the cell membrane is a dynamic structure vital for protection, transport, and communication. Transport mechanisms, passive and active, ensure the cell maintains balance. And if you remember, we had passive transport like osmosis, facilitated diffusion, and diffusion. When we talk about active transport, what is absolutely crucial is to remember the use and need of ATP to make it happen. And active kind of implies that activity is involved, right? And you can remember the mountain metaphor, whereby you're trying to push something uphill. You will have resistance, right? So you need to create and use energy to be able to push against this gradient that is pushing down. This is not happening when we talk about passive transport. And finally then, membranes enable cells to respond to signals and interact with their environment. Now with this foundation, we're ready to explore how cells communicate with each other in cell signaling next. So we have reached the end of chapter 3. Continue now to chapter 4. Welcome to another lecture by MedicoMedics. Cell Biology, Chapter 4, Cell Signaling. In this lecture, we will discuss why cell signaling is important, components of cell signaling, different types, receptors and their types. We will discuss the signal transduction pathways, major signal transduction pathways, regulation of cell signaling, disruptions, and end with a summary. So why is cell signaling important? Well, it plays a role in coordination of activities, so it ensures cells work together for growth, repair, and immune responses. It's important in response to the environment, so cells adapt to change like nutrient levels or stress. Regulation of processes, so controls essential processes like metabolism, cell division, and apoptosis. Clinical relevance. Disruptions in signaling can lead to diseases like cancer, diabetes, or autoimmune disorders. So imagine if a factory's workers couldn't communicate. Nothing would get done. So that's what happens when cells can't signal. Now let's explore the key components involved in cell signaling. So one component is signaling molecules or ligands. These are chemical messengers like hormones or neurotransmitters and cytokines, etc. And if you look at this illustration here, you see the ligand in blue. So any molecule essentially binding to a receptor. That leads us to receptors that are proteins on the cell surface or inside the cell that bind to these ligands. When that has happened, we then move on to signal transduction pathway. That's a series of steps converting the signal into a response. And then we have the target cell response, which could be, for example, activation of an enzyme or changes in the cell's behavior or gene expression, etc. So a ligand binds to the receptor. We have a relay of the signal leading to a response. So you can think of signaling like receiving a message, right? The ligand is the letter, the receptor is then the mailbox, and the response is reading the letter and then acting on it. Now let's examine the different types of signaling cells used in communication here. So types of cell signaling. We have autocrine signaling, where the cell sends signals to itself. Examples: cancer cells producing growth signals. So autocrine is like talking to yourself. Then we have paracrine signaling. This is when signals target nearby cells. Example can be neurotransmitters in synaptic signaling. Then we move on to endocrine signaling, where signals travel long distances via the bloodstream. Examples include hormones like insulin. And then we have direct contact, or juxtacrine signaling, where cells communicate through direct physical contact. Example is immune cells interacting with infected cells. Now let's focus on how these signals are received by the cell through receptors. We have membrane receptors. These are found on the cell surface. They bind large or hydrophilic ligands. If you remember the head, right, we had the cell membrane made of a phospholipid bilayer, right? And we said that the head was hydrophilic, the tails were hydrophobic. So the ligands on the cell membrane are therefore hydrophilic. Examples include G protein coupled receptors or ion channel receptors. So that's membrane receptors. Now we have intracellular receptors. These are found inside of the cell, and these bind small or hydrophobic ligands like steroid hormones. So receptors are like locks, right? And these locks open only if you have the right key, which here is the signals. Now, after this signal has been received, the cell has to process it. So let's see how this happens in signal transduction. Now, the definition of signal transduction is the conversion of an extracellular signal into an intracellular response. Like we said, we had the cell membrane, we had a ligand binding, right? So it's an extracellular, it's outside of the cell. And it's supposed to convert this into an intracellular response. Now, the key steps include, first of all, reception. So the ligand binds to a receptor. That's step one. Or look at this illustration here. Here is step one. Then we have transduction, signal transduction, which is a cascade of molecular interactions amplifying the signal. Right? The process here. And then finally, we will have the response. So specific cellular activity like gene expression or enzyme activation. An example can be epinephrine, right? Epinephrine binding to a receptor can trigger glycogen breakdown in liver cells. So if this was a liver cell and epinephrine binds to a receptor on its cell membrane, glycogen could start breaking down. That's a perfect clinical example of signal transduction pathways. Another pathway is the major signal transduction pathway, or pathways. We have, for example, G protein coupled receptor pathway. It involves activation of G proteins and second messengers like cyclic AMP. Example of this is hormone signaling. Another one is receptor tyrosine kinase pathway. It triggers phosphorylation cascades. Example of this is insulin signaling. Another pathway is ion channel pathway. It opens or closes ion channels in response to signals. Example of this is nerve impulses. And each pathway is like a different road leading to the same destination, which is essentially getting the cell to do something. Now let's see how these signaling processes are regulated to prevent chaos from occurring. So when discussing the regulation of cell signaling, we have signal amplification. Small signals produce large responses, for example, cascade effect. Now we have feedback mechanisms, whereby we either have positive feedback, where the signal is enhanced, or we have negative feedback, which reduces the signal to maintain balance. Finally, we have signal termination, so mechanisms like receptor inactivation or degradation of signal molecules will terminate the signal. So cell signaling can be compared to a thermostat. It can amplify signals when necessary, but turns them off to avoid overheating. But let's look at what happens when signaling goes wrong. So in different pathologies, cell signaling has been disrupted. For example, in cancer, where overactive growth signals, or we have faulty tumor suppressor pathways. It's either one or the other. If it's overactive growth signals, the cell will continuously divide and produce more cells, leading eventually to a tissue, leading to the formation of the tumor. Or we have other pathways that are supposed to signal that these cells are not supposed to be dividing. Like we have tumor suppressor genes, for example. If they are not functional, if they are not signaling the way they're supposed to, we won't have the tumor suppressor in place. So this overgrowth can also keep occurring. Another example is diabetes, where we have impaired insulin signaling. We can talk about neurodegenerative diseases, where faulty neurotransmitter signaling occurs in conditions like Parkinson's. And we can mention autoimmune disorders, where overactive immune signaling lead to tissue damage. So, in summary then, when signals misfire or don't get through, the whole system could break down, leading to different diseases. And here we mentioned 4 different types of diseases. So in summary then, cell signaling is essential for coordination, adaptation, and regulation. It involves ligands, receptors, transduction pathways, and cellular responses. Disruptions in signaling are at the heart of many diseases. Understanding signaling pathways provides targets for drug therapy. And hopefully now it starts becoming more and more clear why we study cell biology and why it is so important. Because signaling is an essential part to the function of cells. And as we just mentioned, if these signals are not working as they're supposed to, we could have, for example, overactive growth signals leading to a disease like cancer. Or we could have diabetes, or Parkinson's, or autoimmune diseases, etc., etc. And that's the end of Chapter 4. Continue now to Chapter 5. Welcome to another lecture by MedicoMedics, Cell Biology, Chapter 5, Cell Communication and Adhesion. So in this lecture, we will have an overview of cell junctions, discuss different types. We will talk about the extracellular matrix, integrins and their role, the importance in tissue organization, role in disease, and end with a summary. So cell junctions are specialized structures that connect cells and provide mechanical support and communication. Functions include holding the cells together in tissues, facilitating communication between neighboring cells, and control movement of substances across tissues. So cell junctions are like bridges and tunnels between cells, keeping them connected and working together. Now let's look at types of cell junctions and their specific roles. So we have tight junctions. These seal neighboring cells together to prevent leakage of molecules between them. An example of this is the blood-brain barrier. So as the name implies, it's tight, right? We don't want everything floating around in our blood to enter into our central nervous system. So we have something called the blood-brain barrier, which we will study in other future lectures. Now, we also have gap junctions. These are channels allowing direct communication and passage of ions or small molecules between cells. An example is heart muscle cells for synchronized contractions. And then we have desmosomes. So these provide mechanical strength by anchoring cells together. Example, skin cells to resist tearing. Furthermore, we have adherence junctions connecting actin cytoskeletons of the neighboring cells. An example is tissue remodeling during development. So here you have an illustration of tight junctions. To remember it, just remember the word tight. Another one is gap junctions. And then we have the desmosome. So let's move on to the extracellular matrix. It is a network of proteins and polysaccharides outside the cells that provide structural and biochemical support. It's made of collagen, which provides tensile strength. We find elastin, which offers elasticity, and proteoglycans that retain water and provide hydration, and finally fibronectin and laminin. These help cells adhere to the extracellular matrix. What is the function of the extracellular matrix? Well, it organizes cells into tissues. It guides cell migration during wound healing, and it influences cell behavior through signaling pathways. So the extracellular matrix is like glue, and it's like glue for tissues, holding cells in place and helping them communicate. Now let's look at integrins, the proteins that anchor the cells to the extracellular matrix. So integrins are transmembrane proteins that connect the extracellular matrix to the cell's cytoskeleton. Its functions include, indeed, to anchor cells to the matrix. Second, to relay signals between the extracellular matrix and the cell's interior. And it helps in regulating cell behavior, like growth, migration, survival, etc. Why is it clinically relevant? Well, integrins are targets for drugs and diseases like cancer and inflammation. So integrins are like anchors and messengers, holding cells in place and helping them and keeping them informed about their surroundings. Now let's examine how cell adhesion and communication are crucial for tissue organization. So the importance in tissue organization of these structures. First of all, maintaining tissue structure. So junctions and the extracellular matrix organize cells into functional layers like skin or intestines. Guiding development. So the extracellular matrix directs cell movement during embryogenesis. Its involvement in healing and repair. Cell adhesion helps close wounds by coordinating cell migration. Now, what is its role in disease? Well, if we have a loss of adhesion, so for example, cancer cells lose adhesion to neighboring cells, becoming invasive, so spreading and invading other structures. So we need this adhesion, right? This anchoring to be kept in place. Another one is the extracellular matrix degradation or degradation. So cancer cells, for example, secrete enzymes like MMPs. These break down the extracellular matrix, allowing metastasis or metastasis. And then targeting adhesion in therapy. So we have drugs targeting integrins or the extracellular matrix proteins. These will then be helpful in inhibiting tumor spread. So if we have drugs that can keep this cell anchored to the extracellular matrix, not moving around, it won't be able to go and start invading other structures, leading to chaos, ergo metastasis or indeed cancer. So in summary then, cell junctions ensure structural integrity and communication between cells. We mentioned tight junctions, right? As the name suggests. It's quite difficult getting through. The other one is gap junctions, like in our heart cells. Another one is desmosomes, where cells are kept connected. Then we have the extracellular matrix, which provides supportive structure support and an interactive environment for these cells. We have integrins, which connects the cell to the extracellular matrix and help in relaying signals. If there are disruptions in adhesion, it will contribute to the disease or diseases like, for example, cancer. So, in summary then, cell adhesion and communication are the glue that holds our body together. It keeps our cells connected, informed, and working together in harmony. An easy way to remember these things is to constantly remember what happens if these structures that you are learning now are disrupted. If signals are disrupted, it can overfire, leading to the production of multiple, for example, cell division. So you put this under the umbrella of cancer, and you connect this bit to signaling issues. Another one is the communication between cells, the relaying of signals, which is supported by the extracellular matrix. And these cells are anchored into a place here by the integrins. Play with the idea that these integrins are not functional the way they're supposed to be. So the cell's adhesion to the extracellular matrix will be lost. And indeed, these cells can then move and invade other structures. Again, we are back at cancer. And that's the end of chapter 5. Continue now to chapter 6. Welcome to another lecture by MedicoMedics. Cell Biology, Chapter 6, Cell Cycle and Division. In this lecture, we will discuss the importance of the cell cycle, stages of cell cycle. We will mention mitosis, the steps of cell division, regulation of the cell cycle, disruptions, and then compare mitosis with meiosis. And finally end with a summary. Now, what is the importance of the cell cycle? It's central in growth and development, so it enables organisms to grow from a single cell to trillions of cells. Its involvement in tissue repair and replacement, so it replaces old or damaged cells. Reproduction. For single-cell organisms, division is a way to reproduce. Furthermore, what is the clinical relevance? Well, dysregulated cell cycles can lead to diseases like cancer. Now let's explore the stages of the cell cycle in more detail. When we talk about the stages of the cell cycle, there are some new words that are all very high yield. As a student, you need to repeat these sections a few times to make sure that you remember these orders. So first we have the interphase, which is the preparation for division. And it consists of the G1 phase, or gap 1 phase. Here, cell grows and performs its normal functions. Then we have the S phase, or synthesis, where DNA is replicated. Then we arrive at the G2 phase, where cell prepares for division by making proteins and organelles. So looking at the cell cycle illustration here, we have the G1 phase, then we have the synthesis phase, we have the G2 phase, where it's preparing for division, and then we arrive at the M phase, also known as the mitotic phase. This is where the cell divides into two identical daughter cells. So it includes mitosis, nuclear division, and cytokinesis, cytoplasmic division. So think of the interphase as the cell getting ready for a big event, and the M phase as the main event, where it splits into two. Now let's take a closer look at the events of mitosis, the most dramatic part of the cycle. So mitosis, steps of cell division. Now, just like we discussed the different steps in the interphase, in mitosis, we also have several phases. And again, these need to be repeated and studied carefully. First, we have the prophase, where chromosomes condense and become visible. A nuclear envelope begins to break down. Spindle fibers form. So looking at the illustration here, we have the prophase up here. Now we move to the second stage, which is the metaphase. And what is happening here? So the chromosomes attached to spindle fibers are lined up in the middle. So the chromosomes align in the middle of the cell. It's also called the metaphase plate. After this alignment is made, we then move into the anaphase, where sister chromatids are pulled apart to opposite poles by spindle fibers. So here you see chromosomes have become condensed and visible. The nuclear envelope has begun to break down. These spindle fibers are formed. These fibers then come into action. First, the chromosomes are aligned in the middle of the cell, and then they are pulled apart by these spindle fibers to the opposite pole or side. After that has happened, we then enter into the telophase. So the chromosomes now decondense. In the prophase, they condensed. Now they become decondensed. The nuclear envelope is reforming around each set of the new chromosomes. Why are there two sets? Well, we said that in the anaphase, they were split apart into its own side or opposite poles. Now we're starting to see the image of two different cells appearing. So we have now the final part, which is indeed cytokinesis. And this is where the division of the cytoplasma occurs, making two cells. A mnemonic I myself used in med school. I just read it backwards, and it kind of worded out stamp. And I thought, well, when we're talking about cell division and we're talking about diseases like cancer, etc., the word approval comes to mind. And you will learn in more detail going forward that during the cell cycle, there are checkpoints at place that allow for this one phase to enter into the next one. So it's constantly about approving something so it doesn't mutate into cells we don't want. And this is where you need a stamp of approval. And you just read it backwards. You get prophase, metaphase, anaphase, telophase, and cytokinesis. Maybe it's helpful to you too. And then we arrive indeed to the regulation of the cell cycle. So just like I mentioned a second ago, we have checkpoints in the cell cycle. So we have at G1, we have the G1 checkpoint, which ensures the cell is ready to replicate DNA. So we have here a checkpoint. After that, when it's approved, it goes into synthesis, right? After its synthesis, it arrives at the G2 checkpoint. And this is to ensure that the DNA that has been replicated was successful. And once that has happened, we then move into the mitotic phase, and we have the M checkpoint. And this ensures that chromosomes are properly aligned. Now, we can also mention regulatory proteins. So cyclins and cyclin-dependent kinases control the progression of the cell cycle. We also have tumor suppressor genes, which are there to halt the cycle if errors are detected. Now, what happens when these regulatory mechanisms right? What if they fail? Let's explore a bit disruptions in the cell cycle. So what we could have is uncontrolled cell division. This would then result in cancer, caused by mutations in oncogenes or tumor suppressor genes. Another is cell cycle arrest. So the failure to progress through the cycle can lead to cell death, apoptosis. What is clinical implication here? Well, chemotherapy targets rapidly dividing cells by disrupting their cycle. Remember, if we have cancer, we know that there is an uncontrolled growth happening. We want to stop this. So chemotherapy targets these rapidly dividing cells and disrupting their cycle, leading to a halt in this division. And here we have an illustration, right? So normal division, one cell is divided into two after in cytokinesis. This one cell then enters into the interface again and then goes into the G1 phase, eventually going to the mitotic phase. And inside the mitotic phase, remember stamp, we read backwards. We go through the prophase, we go through the anaphase, etc. Leading to two new cells, and so on and so on. However, when we have cancer cell division, as you can see, it becomes this thing where uncontrolled cell growth stuck together eventually lead to the formation of a tumor. And if this thing then spreads and invades other structures, now we're talking about the cancer has metastasized. So it's invading other structures. It's spreading. Now, when we talk about mitosis, which is central for cell division, we have another one that we're going to now contrast mitosis with, which is meiosis. And meiosis is crucial when we're talking about reproduction. Now let's just briefly compare mitosis with meiosis again. So mitosis produces two identical daughter cells used for growth and repair. Remember, the key thing to remember is that mitosis produces two identical daughter cells. Meiosis produces four non-identical gametes, so sperm or egg. It also reduces chromosome number by half for sexual reproduction. Now you can look at this illustration here where it says mitosis 2n divides into 2 and then that divides into 2. But in meiosis, you see 2n here dividing into 1 and 1, etc. Now what does this 2n mean here? Well, 2n here represents the diploid chromosome number in a cell. Diploid means the cell contains two sets of chromosomes, one set inherited from the mother and one from the father. So in humans, 2n is equal to 46 chromosomes, 23 pairs of homologous chromosomes from each parent. Now, what does the N mean exactly? Well, the N represents the haploid chromosome number, meaning a single set of chromosomes, so no pairs. So in humans, N is 23. Or in 23 chromosomes. And haploid cells are found in gametes, so sperm and egg. And these are produced during meiosis. So the N here is 23 chromosomes. When we do the 2, we do simply, it means 2 multiplied by this, so 46. Now, that was about meiosis. Now, what about 2N in mitosis? So for a moment, imagine we're using the same illustration. So mitosis. So the parent cell, a diploid cell, 2N, right, undergoes mitosis to produce two daughter cells that are also diploid. So 2N, 2N. Compared to meiosis, where 2N became 1N and 1N, meaning 23 chromosomes and 23 chromosomes. Here we still have 46 and 46. Now the chromosome number therefore remains the same. So key points here is mitosis produces two genetically identical cells with the same chromosome number as the parent cell. This is the parent cell here. So because these are quite important concepts and very high yield during your medical exams and studies, it is worth repeating one more time that the key difference between meiosis and mitosis lies in their purpose and outcomes. So mitosis results in two genetically identical diploid daughter cells for growth, repair, and asexual reproduction. We're talking about this side. While meiosis produces four genetically unique haploid cells or gametes for sexual reproduction. Mitosis involves one round of division, maintaining its chromosome number, while meiosis involves two rounds of division. We have meiosis 1 and then we have meiosis 2, reducing chromosome number by half and introducing genetic diversity through processes like crossing over and independent assortment. So in summary then, the cell cycle ensures proper growth, repair, and reproduction of cells. Mitosis and cytokinesis divide a cell into two identical cells. Regulation of the cell cycle prevents errors and diseases like cancer. So understanding the cell cycle is crucial for medical advances, including cell or cancer treatment. Excellent. That's the end of chapter 6. Continue now to chapter 7. Welcome to another lecture by MedicoMedics, Cell Biology, Chapter 7, Genetics and Molecular Biology. In this lecture, we will discuss the structure of DNA, talk about DNA as a genetic blueprint, replication. We will discuss transcription, making RNA, and translation, making proteins. Briefly talk about regulation of gene expression, mutations and their effects, a brief overview of epigenetics, and end with a summary. Now, what is the structure of DNA? It is made of a double helix, so two strands twisted into spiral shape. You can see that clearly in this illustration. So we have one here and the other one here. Now, what are the building blocks? Well, we call them nucleotides. These have a phosphate group. They are made of sugar or deoxyribose, nitrogenous bases or nitrogenous bases. So adenine, thymine, cytosine, and guanine. We have a base pairing rule when we talk about the structure of DNA. So A pairs with T, meaning adenine pairs with thymine, cytosine pairs with guanine, and they are held together by hydrogen bonds. So here is an illustration of a normal DNA sequence. Again, cytosine binds with guanine and adenine with thymine. And here on the right, you have the nitrogenous base illustrated here and here. The minor groove of the helix is here. The major groove is found here. And then we have the sugar phosphate backbone. So the phosphate and the sugar molecules form the structural framework of the DNA, while the nitrogenous bases store and transmit genetic information through specific pairings. This interplay of structure and function allows DNA to serve as a stable yet versatile molecule for encoding life. So DNA as a genetic blueprint. We have genes that are segments of DNA that code for proteins. And we have another term, which is genome, which is the entire set of DNA in an organism. Now, the function is that DNA stores, transmits, and expresses genetic information. Proteins that are made from these genes determine our traits like eye color or our blood type. So DNA is like a recipe book, with each gene being a specific recipe for a protein. Now let's explore how DNA is copied during cell division to ensure genetic continuity. So DNA replication, the purpose is to produce identical copies of DNA for daughter cells during cell division. The steps include unwinding, so DNA strands separate. Base pairing is step 2, so new nucleotides pair with exposed bases. And then we have sealing, so DNA polymerase forms two identical DNA molecules. So if we do a step-by-step brief explanation of DNA replication, we start with the unwinding of the DNA. So helicase, the enzyme, unzips the DNA by breaking the hydrogen bonds between base pairs, creating a replication fork. We then have single-stranded binding proteins, or SSBS, that stabilize these unwound strands. In step 2 then, so we have the unwinding, which was step 1. We then enter step 2, so RNA primer formation. So the enzyme primase lays down an RNA primer. It's a short RNA sequence to provide a starting point for DNA polymerase. Further down, we have leading strand synthesis. So the DNA polymerase now adds nucleotides continuously in the 5 to 3 direction, following the replication fork. And the replication fork was created in step 1. We then have step 4, lagging strand synthesis. Right? On the lagging strand, which runs 3 to 5, DNA polymerase synthesizes Okazaki fragments discontinuously in the 5 to 3 direction, each starting from an RNA primer. After that, we enter step 5. So we have primer removal and replacement. So RNA primers are removed by DNA polymerase 1 or similar enzymes and are replaced with DNA nucleotides. And then finally, we arrive at step 6, which is sealing the gaps. Remember here, sealing the gaps, where DNA ligase joins the Okazaki fragments on the lagging strand, sealing the sugar phosphate backbone. Now, DNA is not the final product. Let's see how cells use DNA to make proteins. Now, here is something we call the central dogma of molecular biology. So the flow of genetic information is DNA to RNA to proteins. Also illustrated here, we have the DNA here, we arrive at RNA and then to proteins. And this is the end result, the thing we want. Now we have two very high yield and important terms here for you to pick up on. One is transcription. That's when DNA is copied into RNA. So from this step to this step is transcription. We then have the second one, translation, where RNA directs the synthesis of proteins. Now, important here is proteins carry out most cellular functions, like enzymes, hormones, and structural components. So proteins are indeed important. So to give you an idea or a metaphor, think of DNA as the master plan, right? And RNA as the messenger of this master plan. It would then make the proteins as the workers building the cell's structures. Now let's look at transcription, which is the first step of this central dogma. Now, transcription, the making of RNA. The purpose of transcription is to create a single-stranded RNA copy of DNA. Steps include initiation, so RNA polymerase, the enzyme, binds to DNA at the promoter region. The second step is elongation, so RNA polymerase adds complementary RNA bases. And finally, we have termination. So RNA polymerase stops at a termination signal, releasing the RNA. Now, we talk about different types of RNA. We have mRNA, which is messenger RNA. Messenger RNA carries the genetic code. We have tRNA, so transfer RNA. These help in assembling proteins. And then we have rRNA, or ribosomal RNA, which forms the ribosomes. And if you remember, ribosomes are protein factories. So transcription is like copying a recipe from a cookbook. So the book, right, the cookbook with all the recipes is DNA. And transcription is like copying this onto a piece of paper and taking this piece of paper into the kitchen. This is transcription. The next step then is translation, where RNA instructions now are used to start cooking proteins. So translation, making proteins. The purpose? To convert messenger RNA into a sequence of amino acids, meaning proteins. The steps here are the same. So we have initiation. mRNA binds to a ribosome, and the first tRNA or transfer RNA pairs with the start codon. Then we have elongation. The ribosome reads the messenger RNA codons, and transfer RNA brings amino acids to build the protein. And then finally, we have termination. The process now stops at a stop codon, and the protein gets released. And we also have some regulation of gene expression. So why is regulation important? Well, it ensures proteins are only made when needed. Some mechanisms of regulation include transcriptional control, so turning genes on or off, post-transcriptional control, modifying RNA, post-translational control, activating or deactivating proteins. So gene regulation is like controlling a music playlist, playing the right song at the right time and then skipping unnecessary tracks. Next, let's explore what happens when the DNA sequence is altered. So let's talk about mutations and their effects. A mutation is defined as a change in the DNA sequence. So we have different types of mutations, like point mutation, where there is a change in a single nucleotide. Insertion and deletion, so adding or removing nucleotides. We have chromosomal mutations, large-scale changes like duplications or deletions. We also have different effects. So a silent mutation has no effect on proteins. Missense mutations change one amino acid, for example, sickle cell anemia. We have nonsense mutations, so it creates a stop codon leading to a shortened protein. If you remember, the stop codon needed during termination was when the protein was let go to go off and do its function. But if it creates a stop codon, it may make it shorter than it's supposed to be. Then we talk about nonsense mutations. And here you can see a normal DNA sequence, everything is in order, and we have the production of normal proteins. If it's an abnormal DNA sequence, we will have an abnormal protein. Now, what is epigenetics? Changes in gene expression without altering the DNA sequence. Mechanisms include DNA methylation, so we add a methyl group to DNA to silence genes. Another way to do it is histone modification, so loosening or tightening DNA packaging to regulate access. This explains how the same DNA can produce different cell types, and it has big implications for diseases and inheritance. So epigenetics is like adding bookmarks to our cookbook, right? Highlighting some of the recipes and hiding others. So in summary then, DNA is the blueprint of life, storing genetic instructions. The central dogma explains the flow of genetic information, DNA to RNA to proteins. Gene expression is tightly regulated and mutations can alter cellular function. Epigenetics adds another layer of control over how genes are expressed. We have now reached the end of this lecture. Continue now to Chapter 8. Welcome to another lecture by MedicoMedics, Learning Made Easy. Cell Biology, Chapter 8, Bioenergetics and Cellular Metabolism. In this lecture, we will cover the importance of energy in cells, present the concept of ATP, the energy currency, do an overview of cellular metabolism, describe glycolysis, the breakdown of glucose, present the citric acid cycle, briefly talk about anaerobic metabolism and the regulation of metabolism, and end with a summary. The importance of energy in cells. So we need energy to power movements, growth, repair. We need it to drive processes like protein synthesis and active transport. Now, the energy currency is ATP, which is short for adenosine triphosphate, and it is the main energy carrier. Now, let's dive deeper into ATP, the molecule that powers cellular activities. So ATP is the energy currency. What is the structure of it? We have adenine, a nitrogenous base. We have ribose, sugar, and 3 phosphate groups. So in the name, you have adenosine triphosphate, meaning 3 phosphate groups. And as you can see in this illustration here, we have 3 phosphate attached to it. Now, how does this generate energy? By breaking the bond of the 3rd phosphate, we release the energy. So ATP becomes converted into ADP, which is adenosine diphosphate, meaning 2 phosphate groups remaining, plus an additional phosphate group. This ATP then is continuously recycled in cells from ADP and from inorganic phosphate to form new ATP. So the synthesis of ATP comes from ADP, so we have adenosine diphosphate, and an inorganic phosphate combining to form ATP. Now, the process of producing ATP requires itself energy, which is provided by cellular respiration or other energy-generating pathways, like some of these we will mention ahead, like glycolysis or oxidative phosphorylation, etc. Now, the high energy bond formed between the 2nd and 3rd phosphate groups is where the energy in ATP is stored. So when the cell needs energy, ATP is broken down or hydrolyzed into ADP and a phosphate. And this reaction is catalyzed, so this falls under catabolism, is catalyzed by the enzyme ATPase. So the energy released from breaking this high energy bond is what powers cellular processes like muscle contraction or active transport, etc. So what is important to remember here is that the energy produced from ATP is not produced from ATP itself. It is rather produced or a product of the breaking of the high energy phosphate bond between the 2nd and 3rd phosphate groups. Now let's explore how cells produce ATP through metabolic pathways. So here we have an overview of cellular metabolism. Now, when we talk about catabolism, we're talking about the breaking down of molecules to release energy. For example, the breakdown of glucose. The other one is anabolism. Here we use energy to build complex molecules. For example, protein synthesis. Now, some key pathways for ATP production is glycolysis, the citric acid cycle, or known as the Krebs cycle, oxidative phosphorylation. So to express it in a different way, to clarify the concept of catabolism and anabolism, is that metabolism itself is like a factory breaking down raw materials, that would be catabolism, and using them to build useful products, that would be anabolism. Now let's start with glycolysis, one of the key pathways for ATP production. Now let's look at glycolysis. Here you see the pathway and a lot of different individual steps described. Now, to make sense of it all, so glycolysis is like breaking a big tree log, which is glucose, right? You can see at the top here, we have glucose. And what glycolysis is essentially breaking this glucose into smaller pieces known as pyruvate. You can see it almost here at the end of this pathway, to release energy. Now, what is missing from this image is presented in the next slide. It's that pyruvate, by the use of the enzyme pyruvate dehydrogenase, can become converted into acetyl-CoA. Now, acetyl-CoA has a pathway into the Krebs cycle, or the TCA cycle. And that's where essentially ATP gets produced. But we need to get from glucose first to pyruvate. So fundamentally, glycolysis is an anaerobic process. Anaerobic essentially means that the process does not require oxygen to occur. And glycolysis is the first step in glucose metabolism, and it occurs, so this bit occurs in the cytoplasm of the cells. So its objective essentially is to break down one molecule of glucose, which is built of 6 carbons, into 2 molecules of pyruvate that have 3 carbons each. And this will lead to the production of 2 ATP molecules and 2 NADH molecules. These are electron carriers. Now, when this has happened, what happens then after glycolysis? So when we have arrived here at pyruvate, we have essentially 2 potential paths to take. Remember, to get here, we did not need oxygen. It's an anaerobic process. But when we arrive at pyruvate, as illustrated here, let me clear up the image. As illustrated here, you have 2 different pathways, one arriving at lactate and one at acetyl-CoA. So if oxygen is available, meaning we have aerobic conditions present, then pyruvate enters the mitochondria and is further metabolized via the citric acid cycle, meaning the Krebs cycle, and oxidative phosphorylation to produce more ATP. So it will then take this pathway of acetyl-CoA into the Krebs cycle. Now, this is happening. This process is occurring in the mitochondria. That's also why I want you to imagine these pathways you see, connecting them to the cell. Many students may just look at this image of a pathway and think of it like texts in a textbook, but you need to consciously think about, what does this mean if I enter into a cell? Where am I? So glycolysis, as we said, the process of breaking down glucose all the way to pyruvate was happening in the cell cytoplasm. Once we are arriving at pyruvate, if we have oxygen available, then we enter into the mitochondria. And this is then, here is where you have to use the illustration of the Krebs cycle and imagine that all of that is happening in the mitochondria. However, before we move on to the Krebs cycle, we just talked about the option of aerobic conditions, meaning oxygen is available, but what if oxygen is unavailable? So what if we have anaerobic conditions present? In that case, pyruvate will be converted into lactate, and the enzyme is lactate dehydrogenase. Now, let's look at the citric acid cycle, also known as the Krebs cycle or the TCA cycle. It is again happening in the mitochondrial matrix. What is the process? If you remember, looking at this cycle, very often you will see an illustration like this. You need to now know that there was an arrow pointing into acetyl-CoA. That was glycolysis. No, that was pyruvate. So essentially, we are saying that there was an aerobic condition here for pyruvate to go into the Krebs cycle. So the process is pyruvate is converted into acetyl-CoA, which enters the cycle. Then it produces NADH and FADH2. These are electron carriers and 2 ATP. They will release carbon dioxide as a byproduct. So the importance of this step is that it supplies high energy electrons for the next stage. So the Krebs cycle is like a power generator breaking down fuel to produce energy-rich molecules. Now, these high-energy electrons move then to the final stage, which is oxidative phosphorylation. So, oxidative phosphorylation, step 3. Now, this is happening in the inner mitochondrial membrane. So, we had glycolysis, right? We took the example of glucose. Going through glycolysis in anaerobic conditions in the cytoplasma. If arriving at pyruvate and we have oxygen available, it is converted into acetyl-CoA, entering the Krebs cycle with the purpose of producing high energy molecules. Now entering into step 3. So we have moved now from the cytoplasma first into the mitochondrial matrix. After that, we move into the inner mitochondrial membrane. Now, what's happening here? So the NADH and FADH2, the electron carriers produced in the TCA cycle, in the Krebs cycle, they donate electrons to the electron transport chain. And these electrons pass through protein complexes, creating a proton gradient. These protons then flow back through ATP synthase, generating ATP. So we have now arrived at the generation of ATP. What is the end result of this process? We will produce the majority of ATP here, which is approximately 34 molecules per 1 glucose. So oxidative phosphorylation is like a hydroelectric dam using the flow of protons to generate a large amount of energy, ATP. And this process, the electron transport chain process, requires oxygen. Oxygen acts as the final electron acceptor. This is also another point worth mentioning, is that this is also the difference between anaerobic and aerobic bacteria, where anaerobic does not have oxygen as a final electron acceptor, while aerobic does. Now, let us ask the question, what happens if oxygen is unavailable? So let's look at anaerobic metabolism. So anaerobic metabolism, the definition is ATP production. So we still produce ATP, but without oxygen. Now the process, as we mentioned, we had first glycolysis, arriving at pyruvate. If we have oxygen, pyruvate enters into the mitochondrial matrix, further into step 3, where we have oxidative phosphorylation, and the end result was 34 ATP. That's a huge amount for one glucose. But if we do not have oxygen, pyruvate is converted into lactate, and the end result will be 2 ATP per glucose. So you can see the difference, whereby one produces 2 ATP, the other one produces 34. So anaerobic metabolism is like running on backup power. It's not as efficient. It's actually way less efficient, but it keeps things going. Now let's briefly mention the regulation of metabolism. So enzymes that are involved in all of these different steps, like pyruvate dehydrogenase or lactate dehydrogenase, etc., 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 these enzymes are controlled by feedback inhibition. So for example, ATP inhibits glycolysis. The point of glycolysis was what? Break down glucose, get it to pyruvate, get it into the TCA cycle, produce more ATP, right? But what if we have a lot of ATP produced? We need to have some kind of system in place where our body receives feedback to stop glycolysis. So the overproduction of ATP will inhibit glycolysis, and this is a feedback inhibition system. And this is what works for enzymes. Another way our metabolism is regulated is by hormonal control. For example, insulin. It promotes glucose uptake and storage. Another one is glucagon. It stimulates glucose release from storage. So metabolism adjusts based on energy demand. Are you fasting? Are you feeding? Etc. Now, glucagon, is worth mentioning, is a hormone released by your pancreatic alpha cells. And indeed, it is to stimulate glucose release from storage. And insulin is also produced or released by your pancreas, but not by your alpha cells, rather by your beta cells. And they promote glucose uptake and storage. So in summary then, ATP is the primary energy carrier generated through glycolysis, the Krebs cycle, and oxidative phosphorylation. 2. Oxygen is essential for efficient energy production, but anaerobic pathways do provide backup. And metabolic pathways are tightly regulated to balance energy supply and demand. Now, from now on, I want you to always visualize these different steps. When we say glycolysis, step 1, the Krebs cycle, step 2, oxidative phosphorylation, step 3. I do not want you, like many medical students actually do, which is they miss the idea to visualize this as it is happening physiologically in our body. So when you hear glycolysis, immediately try to connect it to a cell. Imagine a cell. Imagine the cell cytoplasma. And this breakdown is happening of glucose into pyruvate is happening in the cytoplasma. Now you need to think a step further. You have an organelle here called the mitochondria that you study in your initial courses. Once it enters there into the mitochondrial matrix, this is where we now know then oxygen was present. So we're in the Krebs cycle now. And here these high energy molecules are produced, the carriers needed for step 3. And that was happening in the inner mitochondrial membrane. And when we go to number 2 here, oxygen is essential for efficient energy production. Yes, because this step, right? These steps provided us with approximately 34 ATP, and anaerobic pathways through the production of lactate gave us 2. And that's the end of lecture 8. Continue now to chapter 9. Welcome to another lecture by MedicoMedics, Learning Made Easy. Cell Biology, Chapter 9, Stem Cells and Cellular Differentiation. In this lecture, we will have a brief overview over stem cells. What are they? Types of stem cells, the process of differentiation, applications in regenerative medicine, challenges and ethical concerns, and end with a summary. Now, what are stem cells? Stem cells are undifferentiated cells capable of dividing and specializing into specific cell types. What are the key features of these stem cells? Well, they have the ability to produce identical copies of themselves. So, self-renewal. Furthermore, potency, so the ability to differentiate into other types of cells. Now let's discuss some types of stem cells. First of all, we have the embryonic stem cells. They are found in early embryos. They are pluripotent, which means that they can develop into nearly any cell type in the body. Furthermore, we have adult stem cells found in tissues like the bone marrow and skin. They are multipotent, so they are limited to differentiating into a few cell types. For example, blood cells or skin cells. So immediately you notice a difference between embryonic stem cells and adult stem cells, where embryonic is pluripotent, which means it can develop into nearly any cell type. And multipotent, the adult stem cell, can differentiate into a few cell types. Further down, we have induced pluripotent stem cells. These are reprogrammed adult cells that behave like embryonic stem cells. Now, this revolutionized regenerative medicine by bypassing ethical concerns, and we will return to this subject later. So, embryonic stem cells are like blank notebooks. Adult stem cells are pre-written with some chapters, and induced pluripotent stem cells are edited books returned back to blank pages. Now that we have an idea of the different types, let's see how stem cells differentiate into specific cell types. Now, the process of differentiation. Now, differentiation is the process where stem cells develop into specialized cells with distinct functions. So in this illustration, for example, you can see the stem cell here differentiating into, for example, muscle cells or pancreatic cells, neurons, nerve cells, red blood cells, or epithelial cells, for example. Now, an important question is, what is it that triggers this differentiation to occur? And there are mainly two factions we need to keep in mind here. One is chemical signaling. So, growth factors and hormones, for example, will guide where this cell will end up. Another one is environmental cues, so interactions with neighboring cells and the extracellular matrix. Now, the stages of differentiation include, we start with the stem cell, differentiating to progenitor cells. This progenitor cell then differentiates into a specialized cell, for example, neuron or nerve cell and the muscle cell. So differentiation is like assigning roles in a theater play or in a movie where each cell gets trained for a specific function. Now, what are the applications of this in regenerative medicine? Well, to name a few, we have tissue and organ repair. So stem cells are used to regenerate damaged tissues, for example, heart cells after a heart attack. Another one is stem cell therapy, so bone marrow transplants for blood disorders like leukemia, experimental therapies for spinal cord injuries or, for example, Parkinson's disease. Furthermore, disease modeling and drug testing, so patient-specific induced pluripotent stem cells, are used to study diseases and test new drugs. Organ bioengineering, so stem cells are used to grow organoids, which are miniature organ models for research. Now let's just name a few challenges and ethical concerns. First, there is the scientific challenges. For example, there is the risk of immune rejection in stem cell transplants. There is a potential for uncontrolled growth, leading to tumors. When we talk about ethical concerns, there is a debate over using embryonic stem cells derived from embryos. Now, stem cell research and treatments are subject to strict regulations to ensure safety and ethical practices. So using stem cells is like handling a double-edged sword. They're powerful tools, but they need careful control. So in summary then, stem cells are unique for their ability to self-renew and differentiate into specialized cells. The types of stem cells include embryonic, adult stem cells, and induced pluripotent stem cells. Differentiation is a controlled process guided by signals and environmental cues. Stem cells have immense potential in regenerative medicine, but challenges do remain. That's the end of this lecture. Continue now to Chapter 10. Welcome to another lecture by MedicoMedics, Learning Made Easy. Cell Biology, Chapter 10, Techniques in Cell Biology. So microscopy is the window to the cell. We have different types of microscopy. So we have light microscopy, which uses visible light to observe living or stained cells. It has a magnification up to 1,000 times. Its application is mainly in cell structure study, mitosis, or live cell imaging. And then we have electron microscopes. So scanning electron microscopy we use for surface structures. We have transmission, so internal details with high resolutions. Its application, to study organelles of viruses and detailed ultrastructures. We have fluorescence microscopy, using fluorescent dyes or proteins to highlight specific cellular components. Application, protein localization, cell signaling studies. Now, once cells are observed, they can be isolated and studied further. So let's discuss some cell culture techniques. So cell culture and isolation techniques. Cell culture is like creating a miniature lab environment where cells can grow and be studied. So growing cells in controlled environments like a petri dish or in flasks. Application used in drug testing or stem cell research. Another is isolation techniques like centrifugation, where we separate cell components by density. Another is flow cytometry, so we're sorting cells based on specific markers. And then we can use 3D culture systems where we mimic natural environments for studying tissue behavior. Now, to study cell molecules and interactions, scientists rely on molecular techniques. Let's explore those now. So, among the molecular techniques in cell biology, we can mention, first of all, the polymerase chain reaction technique, or PCR. It amplifies specific DNA sequence for analysis. It's used mainly in genetic testing or detecting mutations. Another one is Western blotting, where we detect specific proteins in a sample using antibodies. Another one is flow cytometry, where we analyze physical and chemical properties of cells. Its applications include immune profiling and cell cycle analysis. So molecular techniques are like detective tools for identifying and analyzing the molecules that make cells tick. Now, you will come across all of these during your medical career or studies, but one that will very frequently appear is the PCR, the polymerase chain reaction test. So let's just give an overview here of how it works step by step. So imagine we're suspecting a patient having the herpes simplex virus. What we do first is we collect a sample. So we take a swab from the sore, right? Patient presents with a sore. We suspect herpes simplex virus. So we do a swab, and what we then do is we isolate the DNA. So the sample is processed to release and isolate the viral DNA. We then prepare the PCR mix. So the isolated DNA is combined with primers to target the herpes virus, DNA polymerase to copy DNA, and other essentials. What we then do is amplify this DNA, so the sample is repeatedly heated and cooled in a PCR machine, where we double the herpes simplex DNA with each cycle. So finally, we detect the virus DNA. So if the herpes simplex virus DNA is present, it is now in high enough quantity to detect, which would then confirm infection. So all in all, we collect a swab, right? We collect the DNA, assuming it's there. We isolate it. We prepare it in a mix. We amplify it, so making it multiply a lot to a level where if it is actually present and it has multiplied, therefore, once it's high enough in quantity, we will be able to detect it and therefore confirm that it was there to begin with. Now, to understand cells in their natural context, imaging technologies play a vital role. So let's discuss them briefly. Now, among the imaging technologies in cell biology, we can mention confocal microscopy, which produces high-resolution 3D images of cells and tissues. Its application is in imaging layers of cells in tissues. Another one is cell live imaging, so observing dynamic processes in living cells, for example, during cell division. Its application in time-lapse studies of cellular processes. Another is super-resolution microscopy. So it breaks the diffraction limit of light for nanometer scale imaging. We're talking really small here. And of course, the application is to study nanoscale structures like protein clusters. Now, let's talk briefly about some applications of cell biology or cell biology techniques. So for medical research, it helps us identify targets for drug development, understanding disease mechanisms like cancer or neurodegeneration. Biotechnology, producing therapeutic proteins like insulin or monoclonal antibodies. In regenerative medicine, studying stem cells for tissue engineering and repair. So in summary then, microscopy reveals cellular structures and processes. Cell culture and isolation allow in-depth study of individual cells. Molecular techniques analyze DNA, RNA, and proteins for insights into cell function. And finally, imaging technologies provide a dynamic view of cellular activities. And that's the end of this lecture. Continue now to Chapter 11. Welcome to another lecture by MedicoMedics, Learning Made Easy. Cell Biology, Chapter 11, Pathophysiology at the Cellular Level. We will here briefly discuss cellular stress responses, mechanisms of cell injury and mechanisms of cell repair, briefly talk about the role of cells in inflammation, and end with a summary. So, cellular stress responses. The definition here is reactions triggered when cells encounter adverse conditions, like toxins or physical damage. Now, we talk about different types of stress. We have oxidative stress caused by reactive oxygen species, or ROS. And these damage DNA, proteins, and lipids. We have endoplasmic reticulum stress, resulting from misfolded or accumulated proteins in the endoplasmic reticulum. We have hypoxic stress, which occurs when cells are deprived of oxygen. Now, what are some outcomes of this? Well, we have, first of all, adaptation. So, for example, an increased antioxidant defense is activated if we are suffering oxidative stress, and we have too much reactive oxygen species damaging our cells' structures. But if prolonged, there could be cell injury or death if the stress is severe or prolonged indeed. So when cells face challenges, indeed they try to adapt, but if the stress is too much, they can get damaged or even die. Now, what is the mechanisms of cell injury? Well, let's talk about some causes of injury. We have physical agents like actual trauma or burns like heat. There are chemicals and toxins that could cause injury, like alcohol or heavy metals. Furthermore, we have infectious agents like bacteria, viruses, etc. Now, the mechanisms of injury then, to name a few, we have ATP depletion. Remember, ATP is our energy currency, main energy currency. So if this is depleted, it affects energy-dependent processes. membrane damage, so it could lead to cell lysis or leakage of contents. Direct DNA damage, triggering repair pathways or programmed cell death, apoptosis. Now we talk about two types of injury. First, reversible injury, where cells recover if the injury is mild. We also have irreversible injury, leading to cell death, either through necrosis or programmed cell death, apoptosis. What are then some mechanisms of cell repair? Well, DNA repair mechanisms include base excision repair, where single base damage is fixed. We have homologous recombination, where repairs of double-stranded breaks are made. Another one is protein quality control, so unfolded protein response clears misfolded proteins in the endoplasmic reticulum. We also have proteasomes, which degrade damaged proteins. Another mechanism is regeneration, so cells regenerate lost or damaged structures through proliferation. So all in all, cells have repair teams that fix DNA, clear damaged proteins, and regenerate lost parts to stay healthy. So briefly then, what is the role of cells in inflammation? So we have inflammatory signals, where damaged cells release signals like cytokines and chemokines to attract immune cells. We then have immune cell recruitment, so neutrophils and macrophages remove dead cells and fight off infections. But we also have chronic inflammation, where persistent inflammation could lead to fibrosis or diseases like arthritis. So inflammation is like calling emergency services to deal with some damage, but too much of it can actually harm healthy nearby tissues. And throughout your studies, in upcoming lectures in physiology and pathophysiology courses, etc., this will be a key part throughout your medical career, because whenever we face damage, internal, external, whatever the type of injury, we typically come across inflammation. Now, what normally we learn is a positive process. So inflammation is the body trying to heal itself from damage. However, we then learn that inflammation, if it's not controlled or if it's extending, overextending, if it's prolonged, it could lead to the opposite effect, actually harming us, causing damage, destruction, etc. So in summary then, cells respond to stress by activating protective mechanisms like antioxidants or repairing damage. Severe injury could lead to cell death through apoptosis, which means programmed cell death, or necrosis, which is uncontrolled cell death. Cells play a key role in inflammation, attracting immune responses to deal with damage or infection. And that's the end of chapter 11. Continue now to chapter 12. Welcome to another lecture by MedicoMedics, Learning Made Easy. Cell Biology, Chapter 12, Cancer Biology. So in this lecture, we will briefly discuss the hallmarks of cancer. We will discuss oncogenes and tumor suppressor genes. We will compare and differentiate between angiogenesis and metastasis, or metastasis. We will also look at molecular basis for cancer therapies and end with a summary. Now, let's discuss the hallmarks of cancer. So, cancer cells exhibit several defining characteristics known as the hallmarks of cancer. First one is uncontrolled proliferation. So it's ignoring signals to stop dividing. Second one is it's evading apoptosis, meaning it's avoiding programmed cell death. Remember, we had checkpoints in place during cell division, and if our systems recognize that a mutation has occurred that should not be happening, or a proliferation is going on that's not supposed to be happening, our body has mechanisms in place to program the cell to die, the faulty cell to die. But one hallmark of cancer is that it's evading this system. So this uncontrolled proliferation can just continue. Furthermore, as this continues, we have angiogenesis. And this stimulates blood vessel growth to supply the tumor. As this tumor is growing, it needs blood supply. And it's creating blood vessels to grow to supply it with its nutrients. Number 4, immortality, so it can divide indefinitely by maintaining telomeres. And when things become very serious, we talk about invasion and metastasis, which means it's spreading to distant tissues. It's invading to other areas of our body. This is when we say that the cancer has spread. And finally, immune evasion. So it avoids being detected and destroyed by our immune system. So it's not only avoiding cell programmed or programmed cell death on this cellular level, but it's also avoiding our immune system from detecting it and destroying it. Now, oncogenes and tumor suppressor genes. Oncogenes, their normal function is to promote cell growth as proto-oncogenes. However, when they are mutated, they become overactive, leading to excessive cell division. Examples of these oncogenes are the RAS gene or the MyC gene or MyC gene. We then have tumor suppressor genes. Their normal function is to halt cell division or promote reparation or repair. If it's mutated, it loses its function, so it allows for unchecked growth. Examples of tumor suppressor genes are p53 or RB1. And then we have DNA repair genes. Their normal function is to fix DNA damage. Mutations can lead to genomic instability. So oncogenes are like a gas pedal stuck down, while tumor suppressor genes are like brakes that have failed. So it's quite fascinating how our body normally have these systems in place. But why is medicine a subject of study? Why do we need doctors? Because these systems that normally promote, for example, oncogenes, normally promote cell growth. That's a positive thing. And we call them proto-oncogenes. But if they become overactive, we will have uncontrolled cell division happening or excessive cell division. Luckily, why we're not developing cancer in even bigger numbers is because we also have tumor suppressor genes. And they are there to stop faulty genes, faulty cells from dividing. Or if they are damaged, they promote reparation. So these are quite delicate and fascinating systems to study. Now let's explore how tumors grow and spread to other parts of the body. So we mentioned in the hallmarks of cancer, angiogenesis and metastasis. So angiogenesis is blood vessel formation. Now these tumors that are growing secrete signals, and these signals asks essentially for new blood vessels to grow. because it needs oxygen and nutrients to keep growing. Now, it sounds like a nice thing, something growing and it's supplying itself to grow. But we're talking about a tumor, right? Then we have metastasis, or when we say that the tumor or the cancer has metastasized, we are saying that it has spread now to other tissues. So the cancer cells break away from the primary tumor. So if we say we differentiate between a benign tumor, it has not spread, and then we have metastasis, or when we say that the tumor has metastasized, part of the cancer cells break away. And what they do is they invade the extracellular matrix and travel through the bloodstream or the lymphatic system elsewhere. And what they do then is they will form secondary tumors in other distant organs and areas. So to give an illustration, angiogenesis is like building roads to supply a growing city. And metastasis or metastasis is like invaders traveling to conquer new territories. So let's examine how our understanding of cancer biology has led to targeted therapies. Now, targeted therapies are drugs designed to block specific molecules that are driving cancer. Examples of these include, for example, HER2 inhibitors or HER2 inhibitors for breast cancer, EGFR inhibitors for lung cancer. What about immunotherapy? Well, enhancing the immune system's ability to detect and destroy cancer cells. Examples could be checkpoint inhibitors like anti-PD-1 or anti-CTLA-4. Another is angiogenesis inhibitors, where we block blood vessel growth to starve the tumor. Remember, once the tumor begins to grow enough, it now needs blood supply to get nutrients to continue growing. But what we do is we block this from happening. So we let the tumor starve. We also have gene therapy, so correcting or replacing defective genes involved in cancer. So in summary then, cancer arises from genetic mutations affecting oncogenes, tumor suppressor genes, and DNA repair genes. Hallmarks of cancer include uncontrolled growth, immortality, and the ability to invade and metastasize. Also avoiding and evading the immune system and evading programmed cell death are also hallmarks of cancer. So understanding cancer biology has led to advanced therapies like targeted drugs and immunotherapy. And that's the end of chapter 12. Continue now to chapter 13. Welcome to another lecture by MedicoMedics, Cell Biology, Chapter 13, Clinical Applications of Cell Biology. In this lecture, we will briefly discuss the role of cell biology in diagnosing diseases, pharmacological targeting of cellular pathways, personalized medicine, cellular therapies, and end with a summary. So what is the role of cell biology in diagnosing diseases? Well, we have biopsy and histopathology, where we examine cells under a microscope to diagnose cancer, infections, and other diseases. We have flow cytometry, where we identify cell types and markers, for example, in leukemia or immune disorders. Molecular diagnostics, so we use it to detect genetic mutations and infections. For example, PCR for detecting COVID-19. Imaging techniques, so live cell imaging and fluorescence microscopy for visualizing cellular abnormalities. Pharmacological targeting of cellular pathways. So, targeted therapy, drugs designed to interact with specific molecules involved in diseases. For example, HER2 inhibitors or HER2 inhibitors in breast cancer. Another one is tyrosine kinase inhibitors in chronic myeloid leukemia. Hormonal therapies, so target hormones responsive cancers. Example is tamoxifen for breast cancer. Antiviral and antibiotic therapies, where we have drugs that block viral replication or bacterial metabolism. Now, what about the role in personalized medicine? So, personalized medicine is tailoring treatments based on patients' genetic and cellular profile. The application could be in genetic testing, where we identify mutations for targeted therapy. For example, BRCA testing in breast cancer. Pharmacogenomics, where we predict drug response based on genetic markers. Example, CAR T therapy, chimeric antigen receptor T cells, for personalized immunotherapy in, for example, blood cancers. So personalized medicine is like designing a custom suit. It fits the unique needs of each patient. Now let's move on to advanced cell-based therapies that are transforming treatment landscapes. So cellular therapies like stem cell therapy, where we replace damaged tissues or cells with stem cells. Examples include bone marrow transplants and experimental therapies for example, Parkinson's or spinal injuries. Gene therapy, where editing or replacing defective genes occur to treat inherited disorders. Examples include CRISPR technique, used to correct for genetic mutations. Another is regenerative medicine, where we're using tissue-engineered constructs and stem cells to repair damaged organs. Example, lab-grown skin for burn patients. So in summary then, cell biology provides diagnostic tools like biopsy, molecular testing, and imaging to detect diseases. Targeted therapies and personalized medicine leverage cellular pathways for precise treatments. Cell-based therapies, including stem cell and gene therapies, offer hope for previously untreatable conditions. So cell biology is the bridge between science and medicine, helping us diagnose, treat, and even prevent diseases. And with that, we conclude the end of this lecture series. Thank you for listening.